Good afternoon, everybody. Welcome. This is Shellie Harshberger. I will be your webinar instructor today. And today's focus is biology and really shining the spotlight on macromolecules, cells, and pathogens. So I am not going to waste anyone's time. I'm going to dive right in. We do have our new approach where we're trying to get our webinars in right around an hour. So that is our goal today. Also remember that today is content. Today is where we prepare for a practice test. And our practice test then will be Wednesday, this exact time, 3 p.m. Eastern. So today's the content, and then Wednesday we'll put it all to work and see if you remember what we learned today. So I'm so pleased to have everybody here with me. Thank you so much for joining me. Before we get into content, I always like to remind you of the amazing things we have available to help you in your TEAS preparation. As you know, these webinars are totally free. We have lots of them. You've probably seen our schedules all over social media. Please take advantage of those. It's such a great overview to give you an idea of what to expect. Also remember that we know you have a very busy schedule, as we all do, right? So with that in mind, you can rewatch old webinars. That is also a great way to access what is out there. You don't have that interactive moment, but you also have the ability to watch them at your convenience. Furthermore, we have our question bank. Now, this is what I get the most positive feedback on. It's amazing. We have, at this point, it says over 1,500. We actually have over 2,000 questions with rationales. And I actually want to show you this because this is what people get the most excited about. This is just one of the things that is just really one of my favorite tools that we have at your disposal. So I am in our Archer review website, and I am in under T7Q Bank. And I just want to show you how easy this is to make a test. So if we just go to create test, you can pick if you want it to receive instant feedback, if you want it timed. And then you can pick unused questions. That's what I'm going to go with. I've played around with this in the past, and that's why it's showing that I only have 1,867 unused questions, because I played around with it in the past. I'm going to pick, let's say, 45 questions for this practice, and see how we have all of our subjects. So since we are in science right now, I'm going to pick science. And then you do have to pick your subcategories. So for example, today, what are we covering? We are covering cell structure, function, and organization. We are covering macromolecule structure and function, and we're also covering microorganisms and disease. So I'm going to pick those. And then you can see, here's our test. Okay, and so we will pick our answers. Let's just do one, just for fun of it. So which of the following statements are true about the levels of organization in the human body? Select all that apply. Tissues are groups of similar cells that perform a specific function. Organs are composed of different tissues working together to carry out specialized tasks. Cells are the most complex level of organization in the human body. Organ systems consist of multiple organs working together to perform essential functions. And molecules are considered the highest level of biological organization. So I'm going to go with A and D. Let's see what it tells us. Also, notice that it helps us how much time is elapsed. We can flag this, we can pause it. Know that has, and see, look, I skipped one. And I wanted to do that on purpose because I kind of wanted you to see what you would see and what would happen there. So notice that we have our questions here. We have what we did get correct. We have what we're missing. that sort of thing. So the correct answers were A, B, and D. I only chose A and D. You see that? And then it explains to us why the correct answers are the correct answers, what's wrong with the incorrect answers, tells us our learning objectives, and it also gives us, from what I understand, a really good visual that's very similar to the TEAS. And so I think that that's really helpful as well. It tells us how frequently people are getting those correct. And then you have the ability to continue. Okay, so I just wanted to show you that. I just love our QBank. I think our QBank is one of our strongest tools, and we've got a lot of them, but it's a really strong one. So to continue back to our presentation and our webinar for today, do note that we have that question bank available to you. There's a free trial, and then you pick the duration that you need, and the prices are so reasonable. I feel so blessed to work for a company that really gives you an affordable product. And so that depends on time. So then you can determine how much duration of time you need, and you can go from there. The other thing that's really amazing is that we have these on-demand videos. So if you've attended our webinars in the past, you know that our webinars cover a just kind of a nice overview of the topics. But unfortunately, we can't dive in deep. We really need to work on. So let's say that you need to understand how to balance a chemistry equation. We've got an on-demand video. Let's say you need to understand the parts of speech. We've got that on-demand video. In biology, we've got deep on-demand videos for processes like transcription, translation, replication, all those kind of things. So it's amazing. Tutoring. We're going to cover that in just a minute, but if you do need tutoring, there's a QR code there. And then we also have packages, and those are the next two things that I'm going to talk about on the next slide. So our packages here, we have two separate ones. The SurePass gives you that question bank that I just showed you, plus those on-demand videos that I was talking about. And we have over 100 hours of on-demand videos. You can also track your performance, and you can set your goals and your objectives and all that type of thing with the SurePass. Now the SurePass Pro, what you get there is a private tutoring session on top of that. And I'll tell you, we've been getting a lot of those. I'm so excited. I have multiple tutoring sessions scheduled this week, and it is a lot of fun. I was formerly an adjunct instructor and a high school teacher, and so one of the things that I miss out on is that direct interaction. So when I get to interact with you guys like this on the webinars, or when I get to interact with you one-on-one on tutoring, that just makes my day. I'm just telling you, that is one of the things that I miss out on. So please, if you're interested in tutoring, hit us up on that. And as far as tutoring, I'm going to jump this page and come back. You have three of us that can meet your needs when it comes to tutoring. Tammy, she can get you squared away with all of your English and reading needs. Myself, I take care of your science. So anatomy and physiology, biology, scientific reasoning, chemistry. I'm your science person. And then Brandon, he's your math guy. He's going to take care of all of your math needs in preparation for the TEAS. And then lastly, like I had skipped over, we are out there on social media. We are everywhere. So you can find us on Facebook. You can find us on Insta. You can find us on TikTok, and you can also email us. So reach out. We are here to help, and we're here to make your TEAS successful. So I see a few comments here. Yeah, Samari, I was a high school teacher for 17 years, and I also did adjunct instructing at our colleges. I did it for IU University, and I did some dual credit work for my students as well. So they were able to get high school and college level credits. So thanks for asking. All right, without further ado, let's dive in today. So as I said, our focus today is macromolecules, cells, structure and function, and then pathogens. So kicking right off with macromolecules. Oh, I am so sorry, guys. I have us on the wrong one. Here we go. When I came back after I was showing you guys everything, I went to the wrong one. That is my apology. Let me scan right through this real quick. I was in tomorrow's, making sure tomorrow was all set. Okay, so here we go. So for today, as I said, we are kicking off with macromolecules and then cells and microorganisms. Those are our three big topics. So with macromolecules are larger molecules that are made up of monomers, individual units that bond together to make the macromolecules. So when you have those individual units, those monomers, and they bond together, they call them polymers. And that poly, let that be a help for you. Let that word poly kind of help you remember. Poly means many, mono means one. So essentially, a macromolecule is many monomers linked together. Okay, and macromolecules are very, very important to us because they make up who we are. They make up living organisms, and they also make up the food we eat. And so macromolecules are built up for our bodily structures, and they're also broken down to be converted into our fuel, ATP. And that's what we see right here. This is showing us that we have an apple, that's our food, and this is our ATP molecule right here. And it's showing then that we're taking that food and we're converting it to our fuel, which is ATP. All right, so moving right along here. Ooh, I don't like that. My pictures are going away. I'm going to hit refresh here, guys. Sorry, as I had mentioned, the wind here sometimes impacts my internet, and it seems to be impacting the pictures. So we're going to try again here and see if it will let me present with pictures. We always like our pictures. All right, so let's get our drawings erased here. No good having lines on our screen. And let's get through this again then. I'm not sure why our pictures are going away other than the wind. All right, that's better. So our macromolecules, there are four broad categories of macromolecules. We've got carbohydrates, lipids. Lipids are also known as our fats, nucleic acids, and proteins. Our visuals here, you should become familiar with these visuals for the TEAS. They would want you to be able to identify that as a carbohydrate. They would want you to be able to identify that as a lipid. This is a nucleic acid and a protein structure that would look like this. Now, the protein structure is one that they do focus on quite a bit. And be attentive to the fact that there's an amine group, a carboxylic group, an R side chain. It's that R side chain that changes to make it a different protein structure. So that if that R side chain changed, it might go from glycine to lysine or something along those lines. The other thing too that they often focus on in macromolecules and like the structure of them is that proteins are pretty key to having nitrogen as a big component, and they often focus on that as well. All right, so when we talk about each of these, we want to be aware of their monomer unit, their polymer unit, and then just some examples of them. So carbohydrates monomer is the monosaccharide. And again, let that prefix go out. We know mono means many, like so Mari reminded us macro means large. Let those prefixes help you remember what it means. So a monomer of carbohydrate is monosaccharide. A polymer of a carbohydrate is a polysaccharide. And you can see these structures are multiple structures linked together, multiple monosaccharides linked together. And then our example here is glycogen in the liver. So glycogen would be a polysaccharide that is in the liver, just kind of waiting to be used for energy when needed. So some other things that you would definitely need to know for the TEAS is examples of monosaccharides, disaccharides, polysaccharides, and so on. So Our number one example of a monosaccharide is glucose. Glucose will get referenced a lot because glucose is the structure that we break down in order to form that ATP or that fuel. Disaccharides, let that prefix di right there help you. Di means two. Di, bi, those both mean two. So this is saying this is two monosaccharides linked together. Okay. And so sucrose is a prime example of that. Okay. Poly means many. So many monosaccharides linked together forms that glycogen. Okay. And glycogen is a storage form essentially of glucose. It gets converted back into glucose when your body needs it. Okay. It's just kind of that storage form of it. And as Samari reminds us out, if you're not on some of the social media pages, you may not see this. She's reminding us that glycogen is kind of glucose. Those two go together because it's converted into glucose. So that's a good time also to let you know that if you're in Zoom, I can see your messages, but no one else can. If you are in any of the other social media pages, you can see each other's messages, just so you're aware of how that works. All right, so lipids. Our next macromolecule that we want to talk about, our monomer for a lipid is a fatty acid. Okay, and the structure that TEAS really focuses on is this visual right here. Fatty acids can vary. They're not as black and white as a carbohydrate. They can vary, but the structure that they really hone in on is this one. A polymer of a lipid would be like your lipid bilayer that makes up the membrane of our cells. And we're going to talk about that lipid bilayer here in just a little bit. So we'll just move right along. And here it is. So phospholipids. So in our cell membrane is a structure that is so vital to our existence because it is what determines what enters and exits our cells. And so it's oriented very uniquely based on its chemical properties. And so let's kind of take a look at this. So first of all, we have what they refer to as our lipid bilayer. That means we have two layers of lipids. So we have our top layer right here, kind of looks like a little dude with a head and two legs. And we have our bottom layer right here, a little dude with two legs. The watery looking lines that are kind of moving is referencing the water that is both extracellular, meaning outside of our cells, and intracellular, inside of our cells. Okay. Someone's asking if steroid would count as a lipid. Absolutely. Very nicely done. So then when we're looking at our phospholipids, as I said, they're oriented based on their chemical structure. And so their chemical structure is that they are actually molecules that have a polar region and a non-polar region, okay? Essentially what that means is their interaction with water is going to be different because of that chemical structure. So the tails are non-polar, okay? Now let these words kind of cue you into what that essentially means for this watery environment that a cell finds itself in. Hydrophobic. Hydro, hydrate, water, okay? Hydro means water. Phobic, think about if you're phobic, if you're afraid of something, that's kind of a way you can pull that together. So you can kind of say that these nonpolar tails are afraid of water. And so because of that, these nonpolar tails orient away from the water, kind of like a magnet that's repelling its equal charge. They're orienting away from that magnet. They don't want to be anywhere near that water. Sorry, sometimes when I do them all creative like that, I hit the screen and it goes back. All right, so our next region, those heads, are known as hydrophilic polar heads. Hydro, water, philic, loving, polar, water is also polar. So they're attracted to one another. So the heads orient towards that watery exterior environment of the cell and the watery interior environment of the cell. Okay. All right, moving right along. Proteins. Now, the monomer for protein is known as an amino acid. And again, you can let some of the phrases or the names clue you in. The amino acid has an amino group, or sometimes they call it an amine group. Let that kind of cue you in. Oh, yeah, that's right. That's the monomer for protein. They have that carboxylic group I talked about earlier. They have a central carbon, and then they have that R side chain. And that R side chain is what changes in order to change the amino acid. The polymer would be a polypeptide. If you see the word peptide, think protein. In biological language, protein and peptide pretty much synonymous. So a polymer of proteins would be polypeptide. They link together kind of like this. And an example of that would be, would be muscle proteins. These would be some of the muscles that would be in the fascia of your muscle tissue. Now, proteins, I always like to shine the spotlight on enzymes because proteins as enzymes, that's one of their most important roles in our body. Okay. What is an enzyme? It is essentially a catalyst that's going to assist chemical reactions without being used up. And that is huge because that enzyme can go on and assist multiple different chemical reactions without ever being used up. Now, what do? They lower the energy required for the reaction to occur. And that's just all it takes. So they just simply lower the energy required for that reaction to occur. Now, what's unique about them is they connect with their substrate. That's the substance that they're connecting with to lower that energy required for the reaction. That's the name of it. They just give it one broad name, substrate, in what they refer to as a lock and key fashion. And that's what we see in our visual here. And why that's important is because while enzymes are incredibly helpful, they are very impacted by temperature and pH. And so if they end up in an environment that is too hot or too cold, they do something known as denature and change their shape, and then they don't work. If they end up in a pH that's too high or too low, same thing, they'll denature and then they don't work. Okay. All right, moving on then to nucleic acids. And I do see I had a question about nucleotides and I was coming to it. So thank you for your patience. So nucleic acids are final macromolecule that we want to talk about. Their monomer is a nucleotide. So again, let that word clue you in. Nucleic acid, nuclei. See how they start the same? So the monomer for nucleic acids is nucleotide. They have three primary segments to them. They have what they refer to as a phosphate group, the sugar, and then the nitrogenous base. And it's this nitrogenous base that will change to change the nucleotide. And the sugar will change depending on if it's DNA or RNA. Now, ATP is actually also a nucleotide. That's something that we don't often talk about because it's a modified nucleotide. What makes it different is that it actually has two more phosphate groups. So if I could put a phosphate there and I could put a phosphate right here. Oops, I don't want to move that. I just want to put a phosphate there. It's too close. It doesn't like it. All right, let's move that out of the way. So essentially it goes phosphate, phosphate, phosphate. Okay, and that is ATP, adenine triphosphate. One phosphate, two phosphates, three phosphates. Okay, and so that's what they're talking about there. Why it's a T. Adenine diphosphate, we break a phosphate off, and that ends up being only two phosphates on there. Examples then I've got here, a polymer of a nucleic acid I have visually here is an RNA. It's a single strand of these nucleotides linked together. Or another example would be DNA. This would be our double helix where we have two strands of these nucleotides linked together. And focusing on DNA is probably the best use of our time here because it is a very important structure and so many other things that you do need to know for biology. So when we first look here, we can spot the nucleotide we were just talking about. So there's our phosphate group, there's our sugar, there's our nitrogenous base. So if we go back, phosphate, sugar, nitrogenous base. So essentially, the sugar and the phosphates, that's what's making the sides of the ladder. The steps, or what they sometimes call the rungs of the ladder, is the nitrogenous bases bonding together. So they just generally use letters to represent them, but here are their names: cytosine, C; guanine, G; adenine, thymine. Now, C and G always pair together. A and T always pair together. Now, the way that I remember that is that we've got our curvy letters, C and G, and our pointy letters, A and T. They're like, they're pointy versus curvy, and that's how they bond together. So someone's asking about what this blue part is called. These are your sugars. So since we're looking at DNA, that's deoxyribose. Okay. All right. Let's see what else. Someone's asking about my voice breaking up. Is anyone else having problems with that? I don't hear it. Again, like I said, it's very possible. It's so windy here today. Sometimes that does impact my internet. So if you're hearing me breaking up, I so apologize. Someone else asked when we were talking about the cell membrane why the tail is nonpolar. That has to do with its chemical structure. So that's more of a chemistry question, but it just has to do with its chemical structure. When we have molecules that bond and they have an even share of electrons, then they'll be nonpolar. If they have an uneven share of electrons, it'll be polar. And that's really the best answer there. Oh, thanks for letting me know you can hear me loud and clear, Samari. And again, if it was breaking up, it could very well be that the wind was getting the better of the internet there. And Instagram's saying they can hear me too. So hang in there, that person who's struggling. All right, so now that we've talked about macromolecules, we want to talk about cells, their processes, and then microorganisms, specifically focusing on pathogens. So I love this visual here. I think this really gives us a full view of what we think about when we talk about cells. And when we talk about cells in this context, our major focus is really eukaryotic cells because those are the cells that make up you and I. But when we're talking about microorganisms, a lot of them do fall into the prokaryotic category. So it's important to understand kind of the distinction between the two. So prokaryotic cells, they actually believe evolutionarily came first on this planet. From an evolutionary standpoint, they were the first cells on Earth. And that word pro kind of stands for like pre almost, like pre-cells, because they're very simple. They don't have what are known as true organelles. They have ribosomes, but that's really it. Their DNA exists in what they refer to as a nucleoid region, but not in a nucleus, not in a protective membrane. And most prokaryotes, or actually all prokaryotes, are unicellular, meaning they're made of one cell. If we go over on the other side and we look at the cells that make up us, they are going to make up multicellular organisms for the most part. There are some single-celled eukaryotes, but they are not the primary group of eukaryotes. Our cells are very complex. We have organelles with designated jobs, such as our nucleus. And we have all of these organelles that are bound inside a membrane that allows them to have a specific, unique job. So eukaryotes then further break down into animal cells and plant cells. Now for the TEAS, I don't focus a lot on plant cells because really the questions are few and far between. But it would be important to just kind of know the difference between our cells and plant cells. So first and foremost, we do not have a cell wall. We have a cell membrane, but we do not have this outer cell wall. That is specific to eukaryotic plant cells. We have mitochondria as our energy source organelle. Plants have mitochondria and chloroplasts. We have lysosomes and centrioles. Plants typically lack centrioles and lysosomes. We have many vacuoles, many small little storage organs or organelles, whereas the plant cell has one very large organelle. And we're going to now take a closer look at the organelles that make up our cells. Someone asked, ribosomes are present in prokaryotes also? Yes. It is the only organelle-ish structure that they have. And then they have that nuclear or not nuclear membrane, they have the nuclear region, but that's it. So we talked earlier about the plasma membrane. And I said it was very important because it is what is going to select what is allowed in or out of the cell. So when I used to teach high school, I used to have a picture of a bouncer standing in front of the plasma membrane because that's how I kind of perceive. It's very selective as to what can enter the cell, what can exit the cell, and all of that exists within a cytoplasm in our cells. This is kind of a jelly-based, water-based liquid that's in our cells, and chemical reactions can occur there. They support the structure of the cell, and it helps hold the organelles in place. So I'm seeing lots of questions out here. Let me take a look here. Yeah, so someone asked, they said the first time they took their TEAS exam, they were asked the different size of micrograms. That would certainly fall into like our scientific reasoning webinar. That would also fall into some of the chemistry questions. So I'm not surprised about that at all. Let's see what else here. And we've got a nice reminder from Samari that cyto means cell. Absolutely. And then let's see. I think we're good there. All right, so now Golgi apparatus. Golgi apparatus is this structure we've got illustrated here, and its job is to process proteins and lipids into vesicles for transportation. So when proteins get made during transcription and translation, which we will talk about, they will then be shipped out to other organelle to be folded and processed and ultimately be transported out of the cell in most cases. And so the Golgi plays a role in that, as well as with lipids. Lysosomes. Now, lysosomes' job are to break down waste and denature proteins. So if you remember earlier when I said that enzymes will denature if they're in the wrong temperature or the wrong pH, well, if they denature, they're going to be sent to lysosomes, and lysosomes will work to break them down so that they can be expelled as waste. Another question looks like we have out there is that cytoplasm is jelly, is a jelly liquidy, jelly kind of substance? Yeah. I mean, it's not pure liquid because then all the organelles would just kind of squish together. But it's not a real hard jelly either. It's kind of like a melting jello, I would say, like a jello that hasn't fully set up yet. Some other comments, the Golgi apparatus can also be called the Golgi complex. That is very true. And Golgi is the only one you will see in a sentence that's capitalized because it's named after a person whose last name was Golgi who had discovered it. All right, so mitochondria. Mitochondria are the organelles in our cells that can convert those macromolecules we talked about earlier into ATP. And remember, ATP is our body's fuel. That is what we need to function. That is what we need to exist. And mitochondria are the stars of that show. And cellular respiration. If you attended one of my last webinars, someone asked for cellular respiration, so I do have a slide on that. So I try. All right, nucleus. Nucleus, I would call the brain of the cell. It is going to control all the actions of the cell, and it's also where our DNA is held. And that is very important because it's an added layer of protection for our DNA. So not only does something need to get into our cell, they would also need to get through the nuclear membrane to impact our DNA. And so it's that added layer of protection. Now, the nucleolus is inside the nucleus. And notice that this is a different structure. I think this is something that we commonly can mistake. So look closely at this word, nucleolus versus nucleus. See how this has Leo in the middle of it? Use that Leo in the middle of it to remember that it's something in the middle of the nucleus. What does it do? It builds ribosomes. All right, we have a smooth endoplasmic. The smooth endoplasmic reticulum is really involved. It's going to make our lipids, it's going to make our hormones, but it's also involved in detoxification. So if we have any toxins in our system, if we have any drugs that it needs to break down, that's the job of the smooth endoplasmic reticulum. Whereas the rough endoplasmic reticulum, its job is really closely aligned with protein synthesis. Protein is our superstar macromolecule. And so rough endoplasmic reticulum is studded with all these ribosomes. And its job is to contain these ribosomes, be responsible for that protein production because of the ribosomes that are on there. Vacuoles. Vacuoles are like the trash bags of our cells. They're going to store fluids. They're going to store some toxins. They'll store some carbs. They are just kind of the catch-all. They're going to store a lot of those different things. All right, microorganisms then. Microorganisms is those often prokaryotic, but not always. But in reference to TEAS, our focus really is on the microorganisms that are pathogens, that are a problem, that cause disease. And that's our next focus. So there are 5 broad categories of microbes or microorganisms. Those are kind of synonyms that we will focus on, that the TEAS focuses on. Microscopic animals, protozoans, fungi, bacteria, and then viruses. So our first one, microscopic animals. Helminths or parasitic worms. So parasitic worms is the general description of helminths. These would be things like tapeworms or roundworms. That would be something that the TEAS might focus on. And they can live in the intestines. And what they will do is they will kind of suck your nutrients away so they can cause malnutrition. And if they get big enough in your intestines, they can obstruct so that food cannot fully pass through there, so that the bolus that has been created cannot fully pass through there. If you have pets, when the vet asks you to bring in a poo sample, this is the kind of thing they're checking for. They will actually take that poo, mix it up with a liquid ingredient to make it a liquid that they can look at under the microscope, and they're looking for these guys. All right, protozoans. Protozoans can also be known as protists, okay? Now, this is one of those rare examples of a single-celled eukaryote, so the type of cells that make up you and I. This is actually the largest group of organisms on the planet, but it seems so weird to think that, but it's true. Now, our example is malaria, and I want to be careful with this because it's not a mosquito. We can see mosquitoes. Those do not need a microscope. We're talking about things that can be seen through a microscope. But malaria is caused by the Plasmodium species of protozoans that infect mosquitoes. And then when a mosquito injects you to take your blood, what they actually do is when they stick their proboscis into your skin, they give you a little bit of a numbing agent so you can't feel them sucking your blood. And within that numbing agent that they've injected in you, they can inject this Plasmodium species that then causes malaria. And that would infect our liver, that would infect our red blood cells, and obviously we know that is a very debilitating, can be deadly disease. Yes, malaria is a protozoan infection, truly. Someone asked that, and that is true. All right, so fungi. So fungi is another one that's kind of odd because there are versions that we don't have to have a microscope for, right? Fungi is a broad category that includes molds and mushrooms and yeast. And molds and mushrooms we can see. But when we think about them in terms of a pathogen, this is where we're looking at two primary things. Something like athlete's foot, and then look closely at this, ringworm. Now we know, if we use logic, fungi is not a worm, but yet there's a disease called ringworm. It's because when it was first discovered, if you look where my cursor is, it looks like a worm underneath the skin. And so they gave it that name and it just kind of stuck. So be very attentive to that. That ringworm is not a worm at all, it's a fungus. But the way it presents itself, it looks like a worm under the skin. Okay. All right. So bacteria. Now, bacteria is something, if you haven't had a lot of experience with yet in your programs in college, you will certainly have it once you get into nursing school. You'll do a lot of bacteria growth on petri dishes and things like that. Bacteria are prokaryotic, so they, remember, belong in that category of the very simple cells. Okay. And they actually have a cell wall. That's something they have in common with plant cells. And because of that cell wall, they can be classified. They can be classified if they have a thick cell wall, they're referred to as gram-positive. If they have a thin cell wall, they can be referred to as gram-negative. And why that's important to you is because many antibiotics target the cell wall to treat that bacterial infection. So now the gram-positive that have that thick cell wall, they are the purple ones. Okay, so gram-positive, purple, gram-negative, pink. Okay, and the color that they present has to do with this the thick wall or thin wall. It's the way they absorb the stain based on their wall thickness. Okay. Now, it is important when we think about bacteria and all these microorganisms we've been talking about them being pathogenic, which they are, but we know that not all bacteria are pathogenic. For example, lactobacillus, which is in our yogurt, is very good for us. We have lots of bacteria that live in and on us. They call it our microbiome, that help us exist, help us fight off bad bacteria, help us have a good balance digestively in our system. So not all bacteria are pathogenic. But we do know there are some, right? We get bacterial infections, we can get food poisoning, we can get meningitis, we can get blood poisoning or sepsis. So those are certainly examples of pathogenic bacteria. All right, so I see lots of questions out there. Let's see, one of them said that they had a question on the TEAS, which plate had the most abundant bacteria. So you would be looking for lots of different colonies on that plate. That's a great question. That's good to know. Thanks for sharing that. Let's see. And someone else talked about how antibiotics are only used for bacteria, not virus. That is so true. And we will talk about that when we get to viruses. Roundworm is caused by a helminth. No, oh yes, my bad. Roundworm is. Let's go back here. Ringworm is fungi, but roundworm would be a helminth. Okay, very good. You guys got great questions today. You're very engaged. Thank you so much for that. All right, so moving on to our next one, viruses. Viruses both terrify me and fascinate me all in one. And the reason that is because scientifically, we can't really even call them a microbe. We kind of loosely categorize them there because we don't know what else to do with them. But they cannot reproduce on their own. They can actually not metabolize on their own. And those are characteristics of life. And so because of that, scientists say, well, I'm going to call them a non-cellular entity. They can't even really say that they're a true organism because they're not. Which is also another reason, like someone had mentioned, that antibiotics are for bacteria, not for viruses. That's exactly true. An antibiotic will not treat a virus at all. Now, there are antivirals, which is kind of new, but viruses really are best prevented via vaccines. So that's one of the things too, as you're preparing for the TEAS and as you're thinking about the TEAS. Not all, you can't say all, but most vaccines are for viral infections. And if you can kind of tie that together, that's another thing to keep in mind. But why I say that viruses kind of terrify me is because what they do is they have their either DNA or RNA that's enclosed in a protein coat, kind of what we have visualized here. And what they do is they attach to our cell, they inject into our cell their own DNA or RNA, and then, like creepy mind control, they completely take over that cell. So whatever job that cell had before, it's done. Now its job is to make viral particles, make more and more and more and more viruses until that cell bursts and all these viral particles go out into your system. So that's why I say they're kind of terrifying, because if this wasn't something that was so teeny tiny microscopic, imagine how scary that would be if it latched onto an organism, completely took it over, and its job was to make lots and lots of viruses. How scary is that? But because it happens on such a small scale, we don't really think about it that way. But viruses both, like I said, fascinate me and kind of terrify me with that concept that they're not living and that they have this ability to hijack our cells. So viruses, we exist with them though every day, right? We know the common cold is a virus. Measles, mumps are viruses, COVID-19 a virus. So we exist with them every day. And there is actually, this was really fascinating. There is evidence, if you do some research, that I think it's like 8% of our DNA is viral DNA. DNA that got injected into our DNA and it just stuck. And then when, our ancestors passed it down to their kids, who passed it down to their kids, and so on. So fascinating, creepy. I could talk all day about viruses, but I certainly don't have time for that, neither do you. All right, so let's see what some other comments we've got out there. Yes, someone said HPV is a virus. Absolutely, and a creepy one that can cause cancer. Do you need to know the different types of bacteria? I would say knowing gram-positive, gram-negative would be an important aspect for bacteria. Samari, I know, I love science too. Thank you for that. And yes, viruses, in order to reproduce, they have to have a host. And that's what's just crazy about them. All right, so we are moving to our last topic today. Our last topic is cellular processes. So our cells have a life cycle. Much like we have a life cycle, we have stages. So, we have newborn, toddler, teenager, adult, young adult, senior citizen, and so on. Our cells also have different stages of their life. And so the longest stage, if we look at our visual over here, the longest stage encompasses this, what they refer to as G1, S, and G2. And they call that interphase. And in G1, the cell is just doing whatever that cell does. It's normal cellular functions. But as it ages and gets into this S phase, it starts going through DNA replication, where it's going to double its DNA. And that's something we're going to talk about, how it does that in just a moment. And then as its DNA is doubled, then it kind of goes through some checks and balances, and it prepares for division. And division is this M in our visual here. That's mitosis. This is where that double DNA gets separated into two separate cells, essentially, that will then break apart in cytokinesis. And so we're going to take a look at each of these things that I've talked about a little more closely. So that first one that we want to talk about is DNA replication. All right, so DNA replication, what's going on here? Essentially, there are two steps. Okay, the first step is we've got an enzyme. Remember, I said enzymes are stars of the show. So we've got an enzyme, DNA helicase, and it's going to unwind our DNA, because remember that DNA is a spiral double helix. And so it's going to unwind it, and it's also going to break the bonds between the bases. So remember, cytosine and guanine are bound, adenine and thymine are bound together, and they're bound by hydrogen bonds. And so this enzyme right here, DNA helicase, this little triangle, goes through and breaks those hydrogen bonds. That's our first step of DNA replication. Our second step of DNA replication is that now that we're unzipped, now we have what I refer to as just simply free-floating nucleosides. So we have adenine floating around, we got thymine floating around, we got cytosine and guanine floating around in that nucleus. And they then are matched to their partners on each of these strands that are now wide open. Okay? And so what happens then is we end up with two new strands of DNA, each with an original strand and each with a new strand. Okay? And what that does is it ensures that both new cells get the exact same genetic information. And in that case, then, we end up with two cells that are completely identical. Okay, and I do want cellular respiration in here, but for some reason it's kind of jumping in front of division. So we're going to move it here. We're going to put it right there. We're going to make that the last thing we talk about. So as we talked about this life cycle that the cell has, we talked about that M phase where it starts going to divide into those two new cells. And so we have mitosis, and we do need to talk about mitosis. This is certainly something that you will need to know about, as well as meiosis. So I've got some more questions and comments out here. So before we dive into that, let's see what we've got here. So someone's asking about, will you be able to review back over the notes, this being your first webinar? So what I recommend you do there is access one of our old webinars over this topic to review that way if you want to review before Wednesday's practice test. That would be the best way to do that. Then you could fast forward, slow down, and really hone in on any topic that you're struggling with. And then replication happens in the S phase. So we're taking a look here. Replication is going to happen right here. Yes, in that S phase. Okay. All right, let's continue then into mitosis. So there are 4 stages of mitosis: prophase, metaphase, anaphase, telophase. There is cytokinesis, but it's actually not a stage of mitosis. It's that final step that splits those two cells apart. And so it is not technically those 4 phases of mitosis. A lot of my students would come up with PMAT for this. I've not heard other little phrases. They just do PMAT to keep them in order: prophase, metaphase, anaphase, telophase. So in prophase, our DNA is condensed into chromosomes. So DNA, when it's hanging out in our nucleus, is referred to as chromatin. It's just kind of loosely hanging out. It's not all tightly wound. But when we get ready to go through division and go through mitosis, it condenses tightly into chromosomes. And then after it's went through division, it consists of 2 identical sister chromatids. Okay, that means this one was just the standard DNA that was there before replication occurred. But then when replication occurred, it got this sister that's identical. And they are joined at the center part, referred to as the centromere. So all of this happens in prophase. So someone's asking the difference between chromatin and chromosome. So chromatin is a very relaxed form of DNA that's just kind of loosely hanging out in the nucleus. Chromosomes are where it winds up really tightly into segmented pairs. All right, so metaphase. If you ever looked at onion root tips in high school, you probably were able to see metaphase. This is where the chromosomes would line up almost in like a perfectly straight line along what would be like the equator of the cell, the center of the cell. And that's why they say equidistance, because it's like the equator or the center of the cell. Then in anaphase, Now we're starting to separate those sister chromatids. Remember, that was copies of the same DNA, identical DNA on each side. And so we want to send one of the chromatids to one side and the other chromatid to the other side, because ultimately our goal is two new cells that are identical. So in anaphase, we're starting to pull them apart. We have spindle fibers that attach to that centromere, and that's what's doing the pulling. Okay, so they're pulling them in opposite directions. Now in telophase, we have almost two new cells. So the chromosomes are now in separate poles, separate ends of the cell. We've got our nuclear membrane that's starting to form. So that's going to be our nuclear membrane around the nucleus. Those chromosomes are going to start breaking back down and getting more comfortable, not being so wound up into chromosomes. They'll start breaking down into chromatin. The spindle fibers break down. We don't need those anymore. And that's the end of mitosis. But it's not the end of cell division. The end result for cell division is cytokinesis. And so cytokinesis, after mitosis ends in telophase, cytokinesis is going to separate them into two genetically identical daughter cells. So this cell matches the original, this cell matches the original. Okay. And the purpose of mitosis is growth and development, replacing old cells, things like that. Someone asked what spindle fibers are. That's a great question. Spindle fibers were these guys right here, these dotted lines that pulled the chromosomes apart. Okay. Someone asked about what they attach to. They attach to the centromeres, the little center things that are holding the sister chromatids together. So these are centromeres, the things that are holding the sister chromatids together in the center. That's what the spindle fibers attach to. You guys are full of questions today. And we have a nice reminder that meta stands for middle, which is why metaphase was the name for this one. So good observations there as well. All right, so meiosis. So up to this point, mitosis was the cell division for every single cell that divides in your body except for sex cell. So I often kind of joke and say mitosis is the process that makes my toes because it's the body cells. It's everything but sex cells. Whereas meiosis, see how it starts off with the word me? I kind of think about how meiosis made me. If it wasn't for sex cells, I wouldn't be here. And meiosis is the process that divides for sex cells. So let's take a look at that. So the broad category of sex cells is called germ cells, which I know can be confusing because germ is also another term we can use for pathogen, but it's not the same. So germ cells is the broad category name for sex cells. Spermatocytes for sperm, oocytes for egg cells. Okay. Now, some things that are important to remember about meiosis is that we get genetic diversity in meiosis. One example of that is through crossing over, and we'll talk about that when we talk about meiosis and its division process. We also get cells, gametes, the broad name for sperm and egg, that have half the number of chromosomes as the original cell. So remember that our cells have 46 chromosomes. But the cells that make up a sperm or an egg end up being only 23 chromosomes. And so it's really good to kind of stop and think, well, why? Why do we need half? Well, we have to think about the fact that if we have a sperm meet up with an egg, and let's say, for conversation's sake, that sperm had 46 chromosomes, and it met up with an egg that had 46 chromosomes, well, now we'd have 92 chromosomes, and we'd really have some problems. So we have to have this process that cuts it in half so that when the sperm has 23 chromosomes and it meets with an egg that has 23 chromosomes, we get to the right chromosome number. Okay. Let's see, what are some other questions? Someone asked if cytokinesis is part of mitosis. No. Mitosis is prophase, metaphase, anaphase, telophase. Cytokinesis is its own little process where it's just splitting what has already almost been two new cells. And I think we're good so far. All right, so moving right along then. Oh, someone else asked a question here. On the TEAS, I got asked what a cleavage furrow is. Oh, that's a great question. Let's go back for a minute. So right here, telophase. This is a cleavage furrow. See how it's like indented between these almost two new cells? That's a cleavage furrow. That's the spot where it will ultimately split to become two new cells. All right, moving right along. Back to meiosis. So we've got this idea we need to have half. So how do we get there? Well, we get there through two divisions. So notice that we have prophase 1, metaphase 1, anaphase 1, telophase 1, and now we go into prophase 2. Okay. Essentially, got to get to 23 chromosomes. Okay, so for the most part, they're no different than mitosis, except we start off with greater chromosome numbers, and then we start getting down to where we started with 46, we now have 23. Okay, now the only other thing that you would want to know is, remember we talked about this idea here of crossing over? Crossing over occurs right here, prophase 1. That is a question you could easily get asked on the TEAS. Where does crossing over occur? Prophase 1. What happens in crossing over is essentially, you could think of your chromosomes as going through Go Fish. They're exchanging DNA, they're exchanging genes. That way, you're not identical to your mother or your father or your brother or your sister. That exchange process occurs there. Now, we don't actually see it, but we do see it in our illustration, but not till we get to anaphase. But it happens right here. And then we start seeing it here. See how the purple has a little orange on it, the orange has a little purple on it, and how that carries on all the way through. So that we end up with 4 genetically unique cells. People are asking what's the difference between the first one and the second one. Pretty much it's all the same, except for prophase 1, we've got that crossing over process. Crossing over has already occurred here, but we have all those same processes occurring. We just have to get from our 46 chromosome number down to 23. That's really our whole goal of this process. All right, so kind of pulling it all together, mitosis versus meiosis. First and foremost, what do they have in common? Well, they're both forms of cell division. They both impact our chromosomes, and they both have to have DNA replication occur first before they can occur. In mitosis, we start with 46 chromosomes, we end with 46 chromosomes. We start with 1 cell, we end with 2. In meiosis, we start with 46 chromosomes, we go through 1 division, we go through another division, we end with 23 chromosomes and 4 genetically unique cells. The type of cells that mitosis divides is somatic cells. Somatic is a broad term for every single body cell except for sex cells. The type of cells that undergo meiosis, germ cells. 2 genetically identical cells in mitosis, 4 genetically unique cells in meiosis, 1 division in mitosis, 2 divisions in meiosis. Okay. All right. Let's see some other things that we've got out there. Recombination is a fancy way of saying crossing over. Absolutely. It's another phrase for it. As far as in meiosis, half the number of chromosomes happens in the second division. Yes. Yes. If we look back here, we still would have 46 chromosomes if we stopped right there. We have to get to that half by going through the second division. Okay. All right. As promised, I threw this in here because someone asked. Cellular respiration, I have not seen in a lot of the documents that are out there for the TEAS, but yet I've seen it in TEAS questions. So we were talking about it in one of my last webinars, and I said, yeah, I'll throw it in there. So cellular respiration, we're kind of jumping out of what we're talking about. It's a cellular process, but it's not a division process. What this is this is the process that occurs in our mitochondria. That's what we have here, that transforms food into energy. That's really what this is. Okay. So some things that you need to recognize: our equation. It's actually a chemical process, and it is focused on glucose ultimately. While yes, we consume other foods, the star of the show is glucose. And so anytime we talk about cellular respiration, the focus is glucose. And it combines the oxygen that we breathe in this equation. So we have glucose plus oxygen that undergo our chemical process, which we will go into detail here in a moment. And then it's water. This actually happens just, and it actually starts this whole process and can continue if there is no oxygen. So what glycolysis does is it breaks glucose into something known as pyruvate, and it produces 2 ATP. Now, as long as there's oxygen present in our body, that pyruvate can then enter into the mitochondria. If there is no oxygen, that has to go into like a fermentation type process, and that's where we get lactic acid buildup, like when we're exercising too much, or too hard, I should say. Now, citric acid cycle, assuming that there is oxygen, would be where that pyruvate's headed. Okay, and that's going to take place in the mitochondria, and it is going to generate what they refer to as electron carriers, NADH, FADH2. And these guys are important for the big payout, the electron transport chain. The electron transport chain actually occurs in the inner membrane of the mitochondria. Okay. And it's going to use oxygen in the mitochondria, and it's going to produce the majority of ATP. Depending on the cell type, it makes about 34 ATP. So you can see why we really want to get the products of glycolysis into the mitochondria, because this is where we're getting our biggest bang for the buck in our electron transport chain. If we're only producing 2 ATP per glucose molecule, we couldn't exist. But if we've got that oxygen and can take it into the mitochondria, then we get that big bang. We get that 34 ATP approximately. Okay? And that's it, folks. How exciting is that? So I always like to bring this stuff back at the end. So those of you that are saying, oh, I'd really like to review before we have that practice test on Wednesday at 3, do this QR code and rewatch some of the webinars. That would be your best review option. But remember, we've got all these QR codes for everything I went over at the beginning. We got our SurePass, our SurePass Pro. We've got all of the places you can find us. I'm Shellie Harshberger. That's who's been coming at you today. And our next one is we try to do them and need some feedback. I would love that in the classroom these days. So if you can give me some feedback, that would just mean the world to me. Aside from that, thank you. Oh, I got some questions. Someone asked, how could you see the slideshow again? You could see it by watching the old webinars. So go into the QR code here, rewatch the old webinars. You can also access those on our Archer webpage. Okay. And yes, Samara, you guys all did great. And thank you again, Samara, you always attended. I love it. So thank you, thank you. But again, I'm going to leave this up. If you could give me some feedback, I would be elated. And you guys, I will look forward to seeing you all on Wednesday. So pat yourselves on the back. Great job today, and we'll talk soon. Take care. Bye-bye.